Okay, and that way we can make this available to others that aren't able to join us. For those that are joining with your computerized devices, you can type in questions that you think of along the way in the little chat box that's within GoToMeetings program. We will have Dr. Lawlor answer questions at the end of the talk. And during the talk, participants will be muted just to eliminate background noise. I am very pleased to introduce our guest speaker today, Dr. Michael Lawlor. Dr. Lawlor is the director of the Pediatric Pathology Neuromuscular Lab at Children's Hospital of Wisconsin, and he's assistant professor in the Department of Pathology and Laboratory Medicine at the Medical College of Wisconsin. Thank you graciously for joining us today. You can go ahead.
Okay, can everybody hear me all right? Okay, everybody else is muted, so I'll take that as a good sign. So first of all, I would like to thank you all very much for inviting me to speak to your group. It's a big honor to be asked by patients and patient families to discuss the work that we do. And in preparation for this talk, I asked Stacy to ask the Nemaline myopathy community about some topics that they would like to discuss, and there were a number of replies. Some of those issues are things that I don't know as much about as other people, but I've tried to put together a presentation that I think might be relevant to the group here, and that also attempts to address within the context of my expertise as many of those issues as possible. As Stacy had mentioned, anybody that has any sort of questions during the presentation can feel free to write them in the chat box, and we can just go through them all at the end of the talk, and hopefully I can give you guys all what you are interested in. So I'm not sure if I need to do this for this talk, but I'm so conditioned on it at this point, and I do it at the beginning of any talk. I do work with a number of pharmaceutical companies, and so I have a few disclosures. I work as a consultant with Sarepta Therapeutics, who are currently in phase 2 and 3 clinical trials with an exon skipping drug for Duchenne muscular dystrophy. I also am supported in part for our research on X-linked myotubular myopathy by sponsored research agreements from Audentes Therapeutics and Valerion Therapeutics. And very recently I started serving on the scientific advisory board for a foundation building strength. So Stacy had mentioned that it might be useful for the audience here to just kind of describe how I became and why I became a muscle disease researcher. So I thought I'd spend a few minutes on that just as a form of introduction. So I've really been interested in muscle disease since I was a kid, actually, and it's been a very important topic to me since about as long as I can remember, into my teenage years at least. I grew up with a close friend down the street who had Duchenne muscular dystrophy. At first I was kind of his babysitter and caretaker when his parents, would go out, and of course, then we became close friends over years, and so that really drove home for me the chronic struggle of dealing with a debilitating muscle disease, and on the course of decades, and by the time Peter passed away when I was in medical school, it was very ingrained in me as an important problem that required anything that could be done should be done to help these kids. And so that really has been a focus of my training and a focus of my research work in terms of trying to get to this point as well trained as possible to start looking into these problems. I did do a medical degree, an MD and a PhD, which is a research degree, both at Loyola University Chicago. My PhD is in neuroscience, and I worked on peripheral nerve disease back then. Then I went to Massachusetts General Hospital to do an anatomic pathology residency and a neuropathology fellowship. And then I finally had the chance to really work intensely in the muscle disease field during a postdoctoral research fellowship, which I did with Alan Beggs at Boston Children's Hospital. And I think Alan's a name that's pretty well recognized in the Nemaline myopathy community. And we worked a lot on myotubular myopathy as well as participating in some Nemaline myopathy projects even early on. And since moving here to form my own lab at the Medical College of Wisconsin and still working closely with Alan, we've continued to work with myotubular myopathy, Nemaline myopathy, and done a few other research projects. And a few years ago, we were also asked to help found a congenital muscle disease tissue repository, which we'll discuss a little bit at the end, to start providing centralized consenting and tissue access for congenital muscle disease research. And that's when Stacy and I started working together, and it's worked out very well so far. So in terms of the talk today, I wanted to talk a little bit about diagnosing Nemaline myopathy. This is probably stuff that most of you guys know, but in terms of putting together the clinical context and why a diagnosis of Nemaline myopathy gets made, and basically as far as that diagnostic process can be pursued in terms of getting a genetic cause, and then talk a little bit about what is known about the potential causes of weakness in Nemaline myopathy, which is based on studies that include animal model studies as well as studies of human tissue that have been analyzed. And then at the end, since this was kind of a popular demand sort of topic, just talk about the possible treatment strategies for Nemaline myopathy. I do specialize in a few sorts of treatment trials, mostly gene therapy and anti-myostatin therapy. So as a result, we'll talk a little bit more about those. The other treatment type studies that have been published, I've got about a slide for each type, and within the context of what I understand about those studies, I'm happy to present them. But the real authors on those studies are more appropriate to really grill with questions if you have. So first of all, in diagnosing Nemaline myopathy, Nemaline myopathy is, of course, one of the congenital myopathies, and diagnosing these can be a little bit of a problem because on clinical grounds you have a lot of symptomatic variability, and Nemaline myopathy is a very good example of this. So when you look at the symptomatology of Nemaline myopathy, there are a variety of different recognized forms of them, each corresponding to a different set of symptoms and signs. But really what's important to notice here is that you have a considerable proportion of them that is severe congenital myopathy, basically from birth. And then you also have some that are adult onset, and you have a fair amount of cases, the majority of cases, occurring in between. And what that practically means is that if you were to go into a neurologist's office or a primary care doctor's office or a geneticist's office and say, I'm weak, it would be very difficult in many cases to predict that you're going to have Nemaline myopathy versus anything else. And this is pretty well recognized. It's pretty common across most of the other congenital myopathies as well. There are certain things like patterns of facial weakness that can help. But in general, it requires a muscle biopsy in order to achieve a definitive diagnosis. And as you can see here, the reason that people get diagnoses of Nemaline myopathy is due to, is this showing up on is the panel here showing up on everybody's screen or not? Yep. It is, okay. Let's see. There we go, okay. So, and this will be due to the presence of these structures which are called Nemaline rods. And you can see these aggregates of rods here. on this Gamori trichrome stain. They stain dark purple. This is actually a preparation for electron microscopy, but it's being viewed at the light microscopic level, and you can once again see these aggregates of material here. And those are all Nemaline rods. And when you look at it on EM, you can see these, they call them electron-dense inclusions, needle-like, or just kind of body-like inclusions. They're very dark, and this is what Nemaline rods tend to look like. And so in the appropriate clinical context, which, as I said, can be extremely variable, if you see a lot of cells with these rods, then you've got a diagnosis of Nemaline myopathy. Now, just in terms of providing a little bit of a disclaimer, Nemaline rods can be seen in other disease states or with advanced age. And so the presence of a few rods on a biopsy or in an inappropriate clinical context doesn't necessarily mean that you're going to have a diagnosis of Nemaline myopathy. So you need to have the correct clinical context and then the presence of these rods to make the diagnosis. Now, following the pathological diagnosis of Nemaline myopathy, many patients will undergo further genetic testing to determine the gene and the mutation responsible. Oh, and this is just a higher power view of what the Nemaline rods look like on EM. And this is comparatively normal muscle here, where you can see the contractile apparatus. These are mitochondria. These triad structures are important for excitation-contraction coupling. And between the myofibrils here, you can see some stored glycogen. And you can see, obviously, this is a much less organized muscle fiber. You can see some of the same contractile organization in some areas. But these rods are a major abnormality, and then there are some abnormalities in terms of how everything else is organized as well. So I guess another piece of background that's useful to know before we go on to the specific genes involved are just the structural basis of what an interference with the contractile apparatus is going to do to muscle contraction. And so really the process of muscle contraction can be summarized a bit by these two figures here. So when you have a muscle fiber, it's innervated by a nerve fiber, and that nerve fiber is going to cause depolarization to stimulate the muscle membrane. And that depolarization provides an electrical current. The electrical current goes down the surface of the muscle fiber, and then it goes into these structures called T-tubules. And then that translates over to this area called the sarcoplasmic reticulum, where there's a lot of calcium stored. And then this causes calcium release into what's called the sarcoplasm, which is just basically the cytoplasmic area of muscle fibers. And in this area, that calcium is going to interact with a variety of things, but what people often care the most about is the contractile apparatus. So here you can see this is what the contractile apparatus looks like. This is a myofibril, okay, which is an overlapping series of thick and thin filaments. And if you look back here, that's what you're looking at right there, okay? Each of these strings of things is a myofibril. And within the myofibril, you're dealing with both thick and thin filaments. Here's the thick filament, here's the thin filament. And the thin filament is produced by the construction of a number of different small proteins, including a bunch of the proteins, as we'll discuss, that are involved in Nemaline myopathy. And the release of calcium allows the thick and the thin filaments to slide past each other gradually, and that's what causes muscle contraction. And so if there's something wrong with this thin filament, you can have weakness. If there's something wrong with calcium release, you can have weakness. If there's something wrong with the thick filament, you could have weakness, and anything in between. Now, we did just talk about this, so I won't dwell on it, but here's a higher magnification view showing the thick and the thin filament. and a variety of these proteins, including nebulin, troponin, tropomyosin, actin, and cofilin. And when you look at the genes associated with Nemaline myopathy, you can see a lot of the same components. So the most common cause is nebulin, then ACTA1, then several tropomyosins, troponin T, cofilin, and then a couple of these Kelch proteins, and then lyomodin 3, which is the most recently discovered one. But the key take-home message is that most of these proteins are known to be a part of this thin filament, and that that thin filament dysregulation of some sort or dysfunction of some sort is likely the central problem in Nemaline myopathy. Now, given that you have a mutation in one of these genes, there's a couple of different types of mutation that one can have. So, and mutations, there's a lot of different jargon associated with specific subtypes of mutation. But in general, you're dealing with either things that cause the protein of that gene to be shortened. So a nonsense or a truncating mutation causes the transcription process to stop early, and that'll cause a shorter protein. But actually what it often causes is destabilization of the protein to the point that it's essentially not there. And so a lot of people think that these nonsense truncating proteins, might just have a shorter protein in there, but in many cases, especially on the animal models, you see that they're undetectable because they just get rooted into defective protein pathways. The other type of mutation we're talking about is missense mutation. And this is not the type of mutation that So, and I should also specify that some of these nonsense and truncating mutations are large insertions or deletions, and that is in contrast to these missense mutations, where you have a change of a single nucleotide, and that's either going to change some structural aspect of the protein, or change how it's spliced, or even add a premature stop site, which would then truncate it. Now, the other thing that deserves talking about in terms of the types of mutations, or dominant versus recessive mutations. And in Nemaline myopathy, you have dominant types of Nemaline myopathy and recessive types. And this partially depends on the function of the flip protein, and it partially depends on what the mutation is doing to it. So a reason that a mutation might be recessive depends on a couple of factors. In general, recessive mutations are those where having half as much of the mutation is still sufficient for relatively normal function, and so that you need to have a more massive decrease in the amounts or the amount of mutated protein to cause a problem. In contrast, some mutations, like many of the ACTA1 mutations, are dominant in that when you produce the abnormal protein, it actually affects the function of the non-mutant actins, and so it'll produce disease in essentially anybody that has that as a single copy of the gene. In contrast, for recessive mutations, you'd need 2 copies of the problematic gene, or 2 problems in that gene, in order to to have the issue. So now to move on from there, we'll kind of integrate these topics as we go through a lot of the treatment and the pathogenesis stuff. There's a lot of potential causes of weakness in Nemaline myopathy, and these have been identified, like I had said, in a number of animal trials, as well as in studies of frozen human tissue, or specifically treated human tissue from biopsies.
Oh, just telling us about an appointment.
So one thing that is proposed as a potential cause of weakness in all forms of Nemaline myopathy is interference with contraction by non-contractile areas. And so this principally talks about the fact that with a bunch of Nemaline rods all over the place, you may not be able to contract as well, and that's certainly a good point. The consequence of many of the studies that have been done, however, shows that this is not the major cause of weakness in a lot of the Nemaline myopathy that's been studied. And that's also supported by the fact that when you look at a patient's muscle biopsy, it's not really possible to determine how symptomatically severe they are based on the number of Nemaline rods that you're seeing. So you could have a patient with comparatively normal function that's absolutely got myofibers that are riddled with Nemaline rods, or you could have a person with relatively low Nemaline rod burden that is very affected. So other things that have been noted are decreased thin filament length as a potential cause of weakness in Nemaline and LMOD3, lack of contractile units in LMOD3, abnormal cross-bridge binding or cycling. So cross-bridge cycling refers to this process of these the myosin and the thick and the thin filaments sliding next to each other to produce contraction. So that can be abnormal in Nemaline, ActRIIB-1, and TPM3. Disruption of interactions between actin and other molecules, which has been documented in ActRIIB, Nemaline, and cofilin. Destabilization of Nemaline and LMOD3, which is seen in KLHL40. Abnormal calcium sensitivity, which is seen in Nemaline and tropomyosin 3. Abnormal muscle repair, which has been seen in tropomyosin 3. And at least one of the animal models of ActRIIB-1 has also postulated some metabolic abnormalities based on some imaging studies. Now, what I should mention here is that these aren't necessarily specific to these subtypes of Nemaline myopathy as causes of weakness, because in many cases there hasn't been a broad spectrum comparison across all of the available mutations. So these represent where it's been demonstrated, but these causes of weakness have not been excluded necessarily in all of the other mutations. So with that bit of background, I think I'd like to spend most of the rest of the time talking about potential therapeutic avenues in Nemaline myopathy. And I can see even from the couple of questions that have popped up in the side that is the primary area of interest here. So we're going to talk a bit about gene-specific approaches. So gene therapy, cell therapy, protein replacement therapy, gene silencing, and gene editing, which the thing that people seem to be talking about with gene editing most now is technology called CRISPR. We'll also talk about mutation-specific approaches, including exon skipping and stop codon read-through. We'll talk about myostatin inhibition, and we'll talk about calcium sensitizers. And we'll also talk a little bit about tyrosine, which looks like I forgot to list here. With some of these things, I'm just going to talk about what they are in general, because I don't specifically work in those areas. And with other things, including the gene therapy and the myostatin inhibition, we'll delve a little bit more specifically into the work that we've done. So with the gene-specific approaches, there's a lot of jargon that's wandering around now, and there's a lot of different technologies that are currently under development. As a person that is kind of centered in the testing of therapies for muscle diseases, my current sense of things is that there's a lot of technologies that are being developed that have very interesting possible utilities, and that we're still, as a field, in the process of mixing and matching the right approaches to the right therapies. And a part of that, of course, is knowing enough about the diseases themselves to convince the people who are developing the drugs that it's worth a shot. So we'll talk a fair amount about gene therapy here, and in general, this usually means using a virus to deliver a normal copy of the mutated gene to the cells, or in this case to the myofibers. In contrast, cell therapy actually uses some sort of form of stem cell to fuse with the muscle, with the abnormal muscle, and it's going to carry in its entire nucleus that should have a normal copy of the gene. And so these are both gene delivery strategies. Gene therapy uses a virus, cell therapy uses stem cells. Protein replacement therapy ends up being one of the strategy where, say that you are missing say that you're missing actin, and then a protein replacement strategy would be infusing some form of actin that could find the myofibers, enter into them, and to go to the correct place. So you're not using your own cells to produce actin or whatever you're replacing. You are directly infusing it and trying to get it to go to the right spot and do the right things. In comparison, when you look at the gene and cell therapy, both of these are strategies to get the correct gene into the cells so that your cells can make the protein that's abnormal or that's missing. There's also technology that is generally called gene silencing, which would be repressing the expression of a gene. So that might be useful in cases of dominant disease where your mutation is affecting the function of something that normally proteins in your cells that would normally be acting much better. And then gene editing is kind of a new concept where you can remove specific areas of DNA and hope that it repairs it more appropriately, or even affect the regulation of gene expression. So you can up or down regulate the expression of an abnormal gene. And so that's got some very interesting potential approaches that are in very early stages of development. Now, just to move into the gene therapy side of things a little bit. So there are some challenges of gene-specific therapies, and I'm going to focus on gene therapies and protein replacement therapies because this is where I really have the most experience here. I have never participated in a cell therapy study, so I don't have as much practical experience in it. But there are some ways in which it's superior to these approaches, and some ways in which it's more limited. So as far as focusing on gene and protein replacement therapies, a real challenge is the large amount of muscle tissue that the body requires. So in a 70-kilogram person, sometimes up to 35 kilograms of it can be muscle. And that means you can necessitate very high doses of the virus or of the protein replacement agent. This then corresponds to needing a lot of money for a given dose, but also when you take something that's a foreign protein and you put a ton of it in the body, it can increase the likelihood of an autoimmune response. And for all of these gene-specific things, the autoimmune response is the side effect that you are most worried about. Now, non-targeted therapy, especially for protein therapy, can cause substantial loss of therapeutic molecules. And so what I mean here is that if you don't have a virus or a protein that goes specifically to muscle, but instead it gets eaten by the liver, it doesn't really do you any good. And so you need to design your agent to go to the tissues that you want, and that will allow you to keep the dosing down. Immune responses are a big problem, okay, because you are trying to produce something in the body that your body's not used to seeing. You're trying to produce potentially the normal copy of the gene, or the normal protein, and the reason for disease may be that your body's never seen that protein before. And so, so that is something that you need to keep an eye on, and that's a lot of what the preclinical and clinical trial testing for these things are focused on, is whether or not the immune response is actually causing the patient to be more sick or sick in a different way. And this can actually occur to both the target molecule or for gene therapy, it can also be an immune response to the virus that you put it in. And then a big question for all of these things is the persistence of the therapeutic response. So if you put a virus in somebody, it's not going to last forever, but it could last a fairly long time. If you put a protein into somebody, it's going to degrade at some point, and you're going to need to replace that. And so that's one of the positives and negatives of choosing one of these therapies over the other. Now, specifically focusing on gene therapy, there are some limitations here. So the best vector, so the best type of virus one would use to get a gene into a myofiber right now is called an AAV. That's an adeno-associated virus. And the limitation on size is fairly small. It's 4 kilobases. Now for myotubular myopathy, that's okay because it's a fairly small gene, small molecule, and so you can carry it. Whereas for something like Nebuline's enormous. So AAV is not a candidate for something like Nemaline because you just can't pack it into the virus molecule. And so you would have to use a different strategy. There's the possibility, as I mentioned, of immune responses to viral particles. So that's another kind of autoimmune or side effect possibility that needs to be evaluated. There is the possibility of adverse effects due to viral integration. Some of the early gene therapies had random integration into the genome. They didn't use AAVs, they used other virus types. But in some of the people that got those, the integration of the virus into the genome worked, but it happened to knock out some sort of cancer-related gene, and so then some of those people developed cancers as a result of their gene therapy. So that ends up being something that, once again, you need to evaluate in terms of safety. You need to design your viruses and your drugs to avoid these sort of things. Something that people may not think about is the fact that if you put gene therapy, if you put a virus into your myofibers, you really can't control the doses of your therapeutic molecules after you get in there. So if you're doing like a protein replacement therapy strategy and you accidentally, say that you're doing it for something like actin, and you accidentally give too much of the actin, you can just wait for the body to get rid of it and then adjust your dosing strategy like you would with any other medication. If you accidentally give way too much actin-containing virus to the point that the cells are producing way too much actin, though, with gene therapy, you can't get it back out, or at least not easily. And so that ends up making appropriate dosing and understanding what the expression of too much of your molecule of interest, those sorts of things are that much more important for gene therapy because you can't fix a well-intended mistake. And as a result of all of these things, there's and some other things that we probably don't need to go into, it's a very difficult regulatory environment for gene therapy. And as a result, there's a lot of proof needed up front for the FDA to support a trial. It's not necessarily a bad thing. As you can see here, there are some fairly substantial safety issues here. And these trials have been done incorrectly by some groups in the past. And so it is a reasonable amount of caution because you don't want to embark on a strategy like this without doing a lot of scouting work. So the poster child for gene therapy at the moment in the congenital myopathy field is myotubular myopathy, and our lab has worked with Casey Childers' lab at the University of Washington, Alan Begg's lab in Boston, and Anna Buch-Bello's lab in France to try to push this forward. We're kind of the pathology people on it, and the various other groups, also including Barry Burns' lab in Florida, have been pushing forward other aspects of this work. And in relation to the use of gene therapy, there's a couple of advantages of starting with myotubularin that made it a good candidate. So first of all, myotubularin is an enzyme, and it's small. Okay, so as we mentioned before, it can be packed into an AAV, and the fact that it's an enzyme rather than necessarily a structural protein indicates that you might be able to get much better function by replacing just a little bit of it. So it's the kind of the difference between replacing a brick versus replacing a bricklayer on a construction project. A little myotubularin could go a long way. And there are some reasons to think that that's the case, one of which is that when you look at heterozygote females for MTM1, so either the carrier mothers of patients with MTM or the mothers of animals in the various experimental colonies, they're not very weak and they don't have much muscle pathology at all. And so even just having half as much myotubularin seems to cause a lot of improvement in comparison to these boys that are totally myotubularin deficient, and they're very, very severely symptomatic, and it's a very lethal disease. There is one subset of mouse with myotubularin deficiency that has just a very, very small amount of myotubularin in comparison to the knockout mouse, which has essentially none. And these mice with a small amount of myotubularin are also much more symptomatically stable than the knockout mice. And so that also indicated to us that even if we weren't restoring 100% of the myotubularin, that bumping up the expression even by a little bit could have a big impact. And then a key advantage of myotubularin versus many other muscle diseases is that the animal models were really excellent, and they had very clear behavioral and pathological phenotypes to the point that you can tell whether something is working or whether it is not working reasonably quickly, and so you don't spend lots and lots of time on something that doesn't end up working. So the first gene therapy study done in myotubularin deficient mice was done by Anna Bush-Bellow's group in France in 2008. This is not a study that we were a part of, but was something we were watching with great interest as we were doing our anti-myostatin work. Now what Anna did is she took an AAV containing an intact MTM1 gene, and at the time this was a type of AAV that didn't distribute itself throughout the body. It just kind of stayed in the muscle that you injected it in. And she injected it into the lower legs of mice with myotubular myopathy. And she either injected the AAV that had it, or she injected saline, which is PBS. Okay, and so when you look at what a normal mouse muscle, so this WT stands for wild type, and that's what you call a normal animal in an animal colony for disease. You can see that you've got myofibers here. You've got peripherally placed nuclei. They are of a certain size. When you look at the myotubularin knockout mice, here you've got some internalized nuclei here. The fibers themselves are much smaller, and they look, they look considerably more abnormal. And when you look after these localized injection, they look more normal again. And when you look at the overall muscle size, okay, this is what normal looks like. This is the disease treated with saline, and this is the disease treated with the virus. And you can see that that's also much more normal. Anna's group did a number of functional tests, and when they looked at specific subtypes of muscle, this is the EDLs, the extensor digitorum longus and the tibialis anterior, you can see that this is what the PBS-treated muscles look like in terms of strength, which was pretty awful. And when you looked at the treated, they much more overlapped with what normal muscle was producing. The next step that in this trial, which was also done in Anna's lab, was changing the AAV type to something that was more systemically available. And what you can see here is that this is the survival rate of the mice that are untreated or that get saline instead of the treatment, and that the wild type, or i.e. the normal mice, and the systemically treated AAV mice didn't die over the course of the 200 days tracked. Okay, and this is based on a single dose of the gene therapy. The body weight also approached normal in terms of the way that the curve developed. They didn't look completely normal. But remember, they were starting off at a bit of a deficit, and their ability to grow normally seems to have been relatively restored. And even looking at 6 months, when you looked at a few behavioral whole mouse measurements, so in terms of spontaneous activity and hind limb grip force, you can see that they're pretty comparable to the normal mice. So this is the normal mouse in the white, the treated MTM mice in the black, and they're pretty comparable. And while those studies were being done, Casey Childers, who used to be at Wake Forest and he's now at University of Washington, was running a pilot trial of the gene therapy in a naturally occurring dog model of myotubular myopathy. And this is work that we're doing the pathology on and continue to do the pathology on. And so as you can see here, this is just what the pathology of myotubular myopathy looks like in a dog. It's fairly similar to what you end up seeing in mice and humans. You get small fibers, and you get a lot of central nuclei, and the dogs are extremely weak. Mitochondria are also localized to the wrong spot You'll see on one of the subsequent slides that I've got here that when you look at these NADH preparations, you usually see a pretty even distribution of mitochondria across the normal cells. In myotubular myopathy, they are abnormal in terms of how they're arranged. In dogs, they tend to form these ring-like formations around a lot of the cells. In humans, they tend to aggregate in the middle. And so Casey, when he started doing this trial, replicated that localized AAV trial that Anna had done, because dogs are so much bigger that you wanted to know that you had the right kind of AAV construction before you generated so much that you were dosing an entire dog. And so what he did is he injected, this is a kind of a pseudo-colored CT scan, he injected one of the muscles in one dog limb with the AAV, and he injected the other dog limb with salt water. This is lactated ringer solutions. That's just a different form of salt water. And then looked at the two legs in comparison to each other on the same dog. And what you can see here is this is a setup for evaluating whether or not the dog is capable of producing a single movement where he pushes down a pedal. They're under anesthesia for this, and then the nerve gets stimulated, they push the pedal, and you generate a curve based on how much nerve stimulation you provide, how much strength is produced. And so there's a normal curve for what occurs in a wild-type dog. This is the dog leg that got salt water, and this is the dog leg that got the gene therapy. So within the same dog, leg to leg, you have a huge difference in function, with the gene therapy looking much more promising. And then pathologically, you can see the same thing where this is normal, this is the saline side, and this is the gene therapy side, and the pathology of it looks much more normal in terms of fiber size, in terms of the number of central nuclei, and in terms of the organization of the triads, which are a critical component of the pathology in myotubular myopathy. All of this gets restored by the gene therapy. Now, the next step on this, just like with the dogs, is systemic administration. And so I'm going to show you a video that summarizes this. So here are examples of these dogs. These dogs are very well cared for. They are in a facility where they get a lot of exercise. You saw that was a normal dog next to its sibling that had myotubular myopathy. Here's another one of the myotubular myopathy dogs at 4 months. They're having a lot of trouble moving around. Here's another dog at 4 months, a lot of trouble moving around. Now, these dogs really don't last for more than another week or so after this. No, this is problematic. I'm surprised. Oh, let's try this again. This is worth doing correctly. If this doesn't work, we'll close this down and open it up in QuickTime or something. So, and the other thing to note is that we didn't design these dogs. These dogs were identified as having MTM and have become the foundation of the large animal work that has been done. Using dogs is necessary for gene therapy because the response of dogs to gene therapy is closer to what you would see in humans. Mice don't really predict how many side effects you'll see, whereas dogs tend to have more significant side effects than humans. And so if you can demonstrate that it's safe in dogs, then the FDA will like that in terms of addressing it in humans. Here is the first of the MTM treated dogs. You can see here, hopefully it catches up to itself. Obviously this dog is pretty functional. Okay, I'm going to back out on this and just show you the regular video on this, because this is probably the most important thing that we're going to talk about today. So here's the first of the treated dogs. He's actually the one that got the worst dose out of the ones that we've treated so far. This is the same age as the animals that could just barely move around in the last couple of videos. You can see that he's very functional. Here are the next two dogs that were treated. You can see that they are very boisterous. And they're very energetic. These got a more even distribution of the dose than the first dog did. This is that first dog a year after he got his one dose of the gene therapy. You can see that he's a little lopsided, and he's showing some signs of weakness. This shows the importance of the dosing and having an appropriate dose because it started to wear off. It did take a lot longer than one might expect, though. That was a year. These other dogs that got a more complete and more universal dose, however, kept doing very, very well, and they continue to do very, very well, and we're probably almost at the 2-year point at this point. And so, you know, this is obviously very exciting, very encouraging, and it's forming the basis towards hopefully going towards human clinical trials in myotubular myopathy. So this is these guys 1 year post-infusion, so that's about 1 year and a couple months of age. So now so hopefully you understand why I went into the other. Make sure you saw that. So in terms of how the dogs look, this is the pathology from the treated dogs. This is what I think is an untreated dog because it looks totally normal. And this is what I think is a treated dog because it has a little more pathology in terms of fiber smallness than the untreated guy. But I can't be sure because I'm actually being kept blinded as we go forward on these. But as you can see, when you are dealing with an animal that I'm under suspicion at least has been treated, these three panels are indistinguishable from each other, really, even to me. And all I do all day is look at things like this as a pathologist. So the ability to correct the phenotype is very substantial, both from a behavioral and from a pathological point of view. Now, a part of the ongoing studies with these dogs in terms of getting things ready for human clinical trials is figuring out, you know, if possible, how long a dose might last, but also what we should be looking for in terms of the dose running out. Because with gene therapy, it's certainly possible to give a second dose, but there are just a number of things that you need to keep in mind in terms of dosing correctly and hopefully avoiding an immune response the second time the body sees this gene therapy molecule. So that's all I'm going to say on gene therapy. I'm going to talk really quickly here about mutation-specific approaches, including exon skipping and stop codon read-through. So for types of disease in which you've got a premature stop, like a truncation mutation, similar to what you might find in a Duchenne muscular dystrophy patient, you potentially can fool the body into missing that premature stop site and produce what may be an abnormal protein, but at least a protein that will work better than something that's been stopped prematurely. And so and this is really being investigated right now in the Duchenne muscular dystrophy population, because the idea is that you would take somebody who has no dystrophin whatsoever and allow them to change how they process the dystrophin gene so that they would have some dystrophin that might be a little abnormal. So that would be changing a Duchenne muscular dystrophy person into a mildly affected Becker muscular dystrophy person. And for those of you that have experience with both of those populations, that is a huge, huge difference. Now, exon skipping tricks the cells into using its own splicing machinery to excise a portion of the RNA that actually has the mutation in it. And as a result, it'll produce a smaller version of the protein than one would normally have, but it wouldn't be as unstable as this truncation product that a severe mutation would produce. Now, stop codon read-through is more of a non-specific thing that you would essentially tell the translation machinery to ignore, or at least increase the likelihood that the translation machinery would ignore that premature stop site and read through and produce the rest of the protein. This is a little bit more non-specific, and there are some variants of antibiotics that people are testing that will that evaluate, the best way to do this. Once again, in muscular dystrophy is the context that I know it in. Exon skipping is much more specific because you're skipping a specific part of the gene, and so you'd need a different agent for each different exon or set of exons that might be affected. So different parts of the gene might require a different agent, and each different agent would require a separate FDA approval process. And this is currently fairly far along in the clinical trial process, at least in relation to some of the more common exons involved in Duchenne muscular dystrophy. Now, something I'm going to switch to now, and this is something that I have more personal experience with, is the myostatin system. And so myostatin is a negative regulator of muscle mass. Okay, and so what that means is that it prevents muscle from growing past the point at which it's useful for a person. So while having huge muscles might look nice and might get you, a sports contract or a Hollywood movie or something like that, it's not really energetically efficient. Having a lot of muscle uses up a lot of energy. It also, tends to correlate with decreased fat reserves. And so the body and evolution has developed a system for fine-tuning the amount of muscle mass that you have, and this molecule called myostatin is one of the ways in which the amount of muscle growth is limited. And the way that this was found was through studies of animals like this exceptionally large cow and this exceptionally large dog. And you can see here that they are very, very heavily muscled. A transgenic mouse that was a knockout for myostatin was developed, and this is an example of that versus a wild-type littermate. They're bigger and they're much more muscular. And then over the past decade and a half or so, there have been a number of pharmaceutical companies that have looked into various myostatin inhibition strategies to produce agents that inhibit myostatin one way or another, or inhibit the activin type 2B receptor, which is or prevent binding to the activin type 2B receptor, which is the receptor for myostatin that starts this muscle inhibition cascade. So if you inhibit these things, then ideally the muscle grows much, much larger. And the drug that we've used over the past 5 or 6 years is ActRIIB-mFC. So this is actually a soluble receptor decoy for myostatin and anything else that binds to this receptor. And it basically soaks up all of the myostatin and any other ligands that are circulating around, prevents their binding at the cell surface, and prevents this cascade from being activated. And so we performed several trials in myotubularin deficient mice, in mouse models of myotubular myopathy, that were not very successful, but they were very instructive for us. They showed us that our ability to produce muscle growth in these animals, which we were able to produce to some extent, didn't correlate with increased strength in those animals. And that sent us chasing after some of the excitation contraction coupling problems in myotubular myopathy, and told us that we really needed to go towards more of a gene therapy or a protein replacement therapy strategy to fix that, and that ended up working pretty well. We did like this drug, though, because in regular mice or in wild-type mice, it was producing a lot of weight, muscle, and strength gain. And we said, okay, well, this sounds like the drug's going to be good for something, but we just haven't found it yet. And so our next target to check it on was Nemaline myopathy, and in part, a foundation building strength funded this work. They've been funding the person that's actually been doing all the animal work. And so here are some preliminary results from our trial. These are dated a couple months back because that's what we're approved to show with people. And so what you can see here is that when in this trial, this is using a mouse model of ActRIIB-1 mutation. So it's the ActRIIB-1 H40Y mouse model. And when you look at, first of all, treated or untreated wild-type mice, so normal mice, no treatment. It's this blue line here. And then you treat these healthy mice with this muscle builder, and they get much heavier over time. Okay? When you look at the untreated Nemaline myopathy mice here, and that's this green line, and then compare them to the treated, you can see that you do get the Nemaline myopathy animals are getting heavier. They're not getting as heavy as the treated wild-type animals are, but they are getting heavier to the extent that they essentially completely overlap with the normal animals that have not been treated. In terms of survival in this model, you can see that the untreated animals have a spontaneous mortality rate that it isn't total over the weeks that have been tested, but it is fairly substantial, starting at about 6 weeks of life, and that treatment prevents that to some extent. I will say that this early data was extremely compelling, and that as we've increased the numbers of animals in the trials, these numbers have creeped a little bit closer together, but it does look like there's some sort of protective effect to the drug treatment. And then as far as strength increases are concerned, there's a very clear strength benefit to the treated wild-type mice. There is not as much of a strength benefit in terms of forelimb grip force or in terms of spontaneous activity in the Nemaline myopathy mice, although part of our problem here is I think that we were checking some of the wrong phenotypes, and part of the problem here is that these mice actually aren't that, in general, aren't that sick. As far as the pathology is concerned, you can see that this is what the wild-type mouse looks like on this Gamori trichrome stain that shows Nemaline rods and other inclusions. Treatment of them makes the fibers bigger. It doesn't make more fibers. In terms of the treated Nemaline mice, you can see that you have aggregates of Nemaline rods and other material here, and those don't go away with the treatment. If anything, here they look a little bit worse, but this is pretty variable, and I think that the most truthful statement here is that the treatment likely makes at least a subpopulation of the fibers larger, which is what accounts for the weight gain, but that it doesn't do anything to the burden of Nemaline rods or any other inclusions. And then when you look at the actual specific muscle masses, you can see that in both the wild type and in the Nemaline animals, once you treat them, all of their major muscles go up. So it's not an effect that's specific to muscle. It seems to a specific muscle. It seems to be diffusely effective across all of the muscles involved, certainly on a structural basis, and we're still in the process of figuring out how much that really equates to better function. Now we're going to go soundly outside of my area of expertise. I'm just going to talk very briefly about a trial that was done using tyrosine therapy by a group in Australia using the same mouse model. They did show a symptomatic benefit in these mice, both in terms of how well they walked around, how good their forearm strength was, and in terms of their pathology. And that this is the only animal study I know about tyrosine therapy, but from that perspective, it looks like it's somewhat effective. there is another study, another type of agent that people talk about called calcium sensitizers or troponin activators. This is mostly work done by Henk Grenzier and Koen Ottenheim's group out of Arizona and out of Amsterdam. I was a part of this study, but not this part of the study. And to be honest, a lot of this type of stuff is over my head. That's what I've got Koen and Henk for. But I guess the take-home message is that they were using isolated myofibers from patients, and they were looking at how their contractile apparatus behaved in comparison to a normal patient. When you look at a Nemaline myofiber versus a control myofiber, how do they react? And then they added within the dish some of this troponin activator and looked at the impact of it. And I think the take-home message here was that these Nemaline myopathy samples, I'm fairly sure this was a Nemaline mutation study. I should know that because the only reason I was on this paper was because I helped them get the muscle, but I'm blanking on it right now. They were finding that there was a deficiency in the Nemaline myopathy muscle in terms of using calcium, and that the use of this troponin activator made it more capable of using calcium at lower levels. Okay, so that to some extent it improved the ability to utilize calcium. And so based on this, that agent and that class of agents has now been funded to be studied by the MDA in transgenic animals with Nemaline mutations, and so and that work is ongoing. But at this point, where it's been proven to be effective is at the kind of single myofiber stage, and now it's undergoing testing in an animal model. But I don't know more about it than that. The early part of that grant was involved in selecting the best animal model to use for it, and I think that's where things are at. Okay, and another recent trial that I had no involvement in, but these are people I also collaborate with. So this is Nigel Lang and Gina Ravenscroft and Kristen Nowak's group in Australia. Did a trial, kind of a trial. It was they bred animals together to overexpress cardiac actin in some of the ACTA1 deficient Nemaline myopathy mice, or I shouldn't say deficient, ACTA1 mutant Nemaline myopathy mice. And as I mentioned earlier, with ACTA1 mutations, a big problem is that the mutant ACTA1 can affect the behavior of the non-mutant ACTA1. So it acts in a dominant fashion. And so one potential therapeutic strategy would be to decrease the dose of the mutant ACTA1 in the cells so that a given normal actin molecule is more likely to interact with a non-mutant actin molecule than it is to interact with one of the problematic ones. And the strategy that they used here was the overexpression of cardiac actin. And in doing that, they, at least in one of the two models that they tested, they had a very significant increase in survival and improvement in body mass and pathology and things like that. And so this was very exciting, and it offers a very interesting potential use. And now I think that it's possible that using some of the gene editing types of approaches that are really kind of hot topics right now may be able to take this from the stage of mate transgenic animal A with transgenic animal B to get improvement in the offspring to something where you can directly manipulate this in sick animals. But that's my understanding of where that work is at. And so I guess for the last part of this, I just wanted to quickly talk about where things are headed. And so, in general, we still need to improve our understanding of Nemaline myopathy, and I think probably in the question and answer period we're going to establish that there's still a lot of work to be done, even to understand about how the different genetic causes of Nemaline myopathy even relate to each other, and how their causes of weakness may be similar or different. And all of this is necessary so that we can choose the right models and the right patients in which to test therapies, because there may be something that works for some people that doesn't work for others, and choosing the wrong patient population may lead people to misunderstand the usefulness of certain approaches. There are a lot of promising therapeutic approaches, and all of these approaches need more work in terms of moving forward for therapeutic trials in both cells and animals. And any of those that are real candidates, then we're going to need to figure out ways to translate successful candidates into human studies. And thankfully, the work that we're currently doing in MTM should at least provide us with a roadmap of how to do that very effectively for Nemaline myopathy by the time that time occurs. There are a few things that, given the audience here, I figured I would talk about ways in which the audience here could help. One of them is increasing the profile of Nemaline myopathy, and I realize that this has been a very big priority of a number of the organizations and patients involved. A big potential limitation for the ability to get funding or the ability to get drug companies developing therapies for Nemaline myopathy is just the fact that people may not have heard of it. And certainly, as I've talked to several drug companies, a lot of what I have been doing is informing them on how it's different from muscular dystrophy, because everybody knows what Duchenne muscular dystrophy does, and a lot of the agents that are currently out are based on the hope that it'll work for that. But some of those agents may actually be much better for Nemaline. And so the higher profile everyone can make Nemaline, the more that people will stand up, take notice, and decide that it's a problem worth solving. The other thing that really helps is becoming organized. So as everybody's been discussing, joining patient registries, sharing your clinical data, centralizing things like tissue access, not necessarily with us, but somewhere where it doesn't get lost and it can be accessed. That's really important, because when you end up talking to somebody that wants to invest their agent or their money or their time in developing a treatment, it's very convincing to be able to say, yes, we know how many people there are, we know where they are, and if something was working, despite the fact that this is a rare disease, you wouldn't have a problem populating your clinical trial. And so that's part of the real value of kind of sharing all of this stuff. And also, if you share your data, it's possible that people will be able to identify aspects of the disease that need to be studied as treatments get developed. And so in terms of how the Nemaline myopathy community can help now, I would say get organized, because this is an attractive thing for companies, and improve disease recognition. An option for the registration data sharing is the Congenital Muscle Disease International Registry and other patient registries. And some of these are, they're variably accessible to researchers. And they allow people to do studies to answer questions about how many patients exist and the characteristics of those patients. Tissue donation to us or to other laboratories. This includes prior biopsy tissue that may just be sitting in somebody's freezer. This is really important in particular for Nemaline, because Nemaline itself is more common than some of the other congenital myopathies, but it's also spread across 10 different genes. And so anything that is done therapeutically is going to need to be done on a gene-by-gene basis for Nemaline. And so big numbers of tissue donations, tissue samples are going to be needed, partially to establish how these different genetic causes are different from each other, but also partially to validate whatever we end up finding using the animals. And then in terms of where we're headed, our laboratory is involved in a lot of different things: gene replacement, enzyme replacement therapy, the tissue repository, the discovery and documentation of new genes or diagnoses, and then anti-myostatin therapy and a few other new therapeutic directions. And all of the goal is to go to human clinical trials in both myotubular myopathy and Nemaline myopathy. And ideally, the myotubular myopathy experience that we're getting right now will help us out a lot as we hopefully go in the same direction in Nemaline. And this is just the people in our lab. So this is me, this is Stacy, this is Hui. She's my primary assistant in the laboratory. Stacy's the manager of the congenital muscle disease tissue repository, and Jenny's our animal technician and also helps out a lot in the lab. And as you can see, we've got a lot of different support here. The Nemaline myopathy stuff that we talked about here today is primarily funded by both Foundation Building Strength and the Clinical and Translational Science Institute of Southeastern Wisconsin. And then the gene therapy is partially supported by the NIH, which also partially supports the Nemaline work, as well as on the sponsored research agreements that we had discussed earlier. And I think that's all I have. So let me see what we've got here. Hello? Yes? Okay. Good. Sorry, I don't have a cam.
I'm going to unmute everyone right now so that we can have some dialogue. As long as there's not going to be too much distraction from background noises, I think that'll work.
Okay.
Oh, so the slide availability, let's see. Yeah, I don't see any problem with providing that by mail.
And there are some people, Mike, that are probably joining us with a telephone and they aren't seeing what the questions are.
Okay.
So we might want to just read them out loud.
Yeah. So it looks like...
I like that, just keeping it simple, especially if you want to keep it to an hour. It'll go to what's most salient for people.
Okay.
And then we can look to see what's consistent amongst the groups and notable within the groups.
So the issue of whether or not the slides would be available by mail, sure, I can make them available. Okay, this looks like... Okay, the gene replacement therapy thing I think we talked about. So whether or not gene replacement therapy works, it looks like it's very promising.
Co-counsel structures.
Myotubular myopathy.
They have an office assistant position.
But
I would put an office assistant in a student leader group.
In Nemaline, I think that it's a little bit harder to implement because of the size of the genes. But it is a direction that I would like to see us go in if we can figure out how to package it correctly. In terms of the treatment options available, how would you ever know which treatment options affect your particular mutation? And I think that this speaks to the value of registering in some of these registries like the CMDIR. People who work on these treatment options want to identify you. They want to know that you exist, and they certainly want to be able to contact you if they have something promising. Medical researchers probably won't have your contact data, but the international registry would, and so it's possible then to talk to somebody like the CMDIR, and then they can send out a mailer to the appropriate people and have you contact them. There's a comment here that tyrosine didn't help at all for some people. That's certainly true. It seems like the response to tyrosine is very variable on an individual basis. Okay, and this seems to be an answer to one of the questions here. Okay, so there's a comment here saying they were told that Nemaline myopathy is an issue of making the protein incorrectly as opposed to not making it at all. That's certainly true for a lot of the Nemaline myopathy mutations. But there are some recessive mutations for Nemaline myopathy where the protein doesn't get made at all, or it gets made at very low levels. And as we were talking about, the therapeutic options that would be available for those two different situations would probably be different. So in terms of, there's a question here about how to check whether or not you're registered. I would just quickly call the whatever registry that you're interested in. So it looks like you're talking about Dr. Begg's registry. If you look for his laboratory online, there will be a contact person. For his lab, it's actually Lindsay Swanson. And ask if you are registered with them, and they will be able to tell you very quickly. Or you can ask Stacy. She can help you with that. So there's also mention of all of the things that the CMDIR requests in terms of medical records and whether or not somebody can help coordinate the efforts to register to the CMDIR. That is certainly possible. I would say coordinate it through Stacy at first, and she can help with that. She helps a lot of people with that. And if it's more complicated than what she can manage, more time-consuming than she can manage, she'll put you in contact with a person that can help. So, okay, does NM impact certain fiber types more than others? Is type 1 versus type 2 more affected? It depends on the mutation that you're looking at. Things like tropomyosin 3 mutations and some of the troponin mutations have a real predilection for affecting type 1 fibers but not type 2 fibers. And then other ones, it affects all comers. So in terms of how MTM therapy correlates to possible NM therapies, from a certain perspective, MTM has characteristics like being small and being an enzyme that don't correspond well to a lot of the Nemaline myopathy genes. And so things like gene therapy, which look very promising for MTM, may not be useful in the same form, at least using the identical strategies for Nemaline myopathy. How MTM therapy going forward really helps with Nemaline myopathy, though, is in establishing the usefulness of a rare congenital myopathy in bringing a new drug platform for muscle disease to market. Okay, because a lot of these drug companies are developing things for Duchenne muscular dystrophy because that's the disease they've heard of before, not necessarily because it's the best disease to test their approach in. And even just looking at the way that entire companies have been founded around getting MTM's therapy into humans and the potential for that to get that company's technology to market much faster, people are taking a much more serious look at the congenital myopathies and whether or not that can help accelerate their drug development programs. From the organizational perspective, people like me are involved in organizing preclinical and clinical trials for these types of diseases. And so the more experience that specialists like myself and Alan Beggs and others get in bringing something from the animal stage to the human stage, I'd like to think that we will do it better and more efficiently each time. And so I think that I'm hoping that we will make no mistakes in the MTM therapy organization, but that's maybe a little bit unrealistic. But I think a goal here is not to make the same mistake twice. And so anything that we do organizationally that crosses over from the MTM study to the Nemaline study, of which there should be a lot, should be happening at a very high level of experience and expertise by the time we get to organizing something for NM. Thank you very much for the comments that appreciate the research.
Thank you so much, Stacy and Mike. This is Patty Mitchell. Thanks so much for all you do. Please keep the great research going, and we will do our best to continue to support and fund you. Thank you.
Thank you, Patty.
Are there any other questions that we haven't gotten to yet? Mike is just reviewing the comments over in the chat window just to make sure there isn't anything that wasn't answered yet. If anyone has any additional questions, you can email them to me after this or find me on Facebook. Most of you know how to find me.
Yes, and Stacy's an ideal way to get in contact with me too. If you guys need anything. It seems like a lot of these are answers to questions. I can tell if somebody's typing. Let's see, is there a way we can get an update on our particular mutation if we have been involved in research? So this depends on where the research is being performed. So with the congenital muscle disease tissue repository, there is kind of a standard disclaimer that there's no, because we can't control the behavior of the people that we would distribute to. We can control whether or not we give a person tissue based on whether or not they have a good idea, but we can't control whether or not they come back in contact about things. And so there's a standard disclaimer that says you will not know where your tissue goes, and you will not know what it causes, what the outcome of it is. Now, practically speaking, this is a pretty small patient population. The number of patients with any given mutation type is a bit relatively easy to track as opposed to a large-scale effort. And so what the tissue repository itself will do is it will provide a quarterly or, depending on how much we distribute tissue, will provide a regular update on the types of research that the tissue repository has supported by giving tissue donations, and a synopsis of some sort or a link to the studies that have produced it. And in that case, if you are, say, a tropomyosin tissue donor, you will know that a certain study does tropomyosin research, and even if your tissue didn't go to it, you'd know that it was applicable to you and you'd be able to keep up with it. But logistically speaking, that's our best workaround on that. And I think that may be it.
Okay. If anything else comes up, if you think of other questions, feel free to email me at any time, and I will get Dr. Lawlor to comment on that, and I can respond to you directly. So if there's nothing else, I just want to thank everybody for joining us today. We really appreciate your participation and, of course, your questions. And if there's anything else that we can do to help, just let us know. And thank you so much, Mike, for doing this for us today.
Okay, well, thank you to you all for attending.